Hi, Miss Nikki here. Welcome to Chapter 25. We're going to talk about gene expression. So we're going to do a little bit of history, and then we're going to walk through replication and transcription and translation. So replication, DNA to DNA, transcription, DNA to RNA, and translation, RNA to a protein. A little bit of history here. I want us to realize that we didn't really know the structure of DNA until about mid-century, so about 1950s. Prior to the 1950s, we knew that there was some sort of genetic material, and we knew some of the qualities, that it was able to store information for development and structure and metabolism, and that it was stable and it could be replicated or passed from generation to generation with high accuracy. The basics of what we understand about genetic information started in about the 20s with Griffith's experiment. So before we talk about what he concluded, come down and look at the images. So you can see that a live bacterial strain, S strain, was injected into the mice and they died. Then they took the inside, the live virus, but no capsule, no outer covering, and the mice did not die. Then they heat killed the S strain, injected it into the mice, did not die. Then they heat killed the S strain, and then they took the R strain, mixed them together, and then injected the mouse, and the mouse died. And they were able to withdraw live S strain from the mice. So somehow this by itself didn't kill the mouse, and this by itself didn't kill the mouse. But when I added these two together, they were able to reassemble and cause infection and cause the mouse to die. So Griffith concluded that some substance must have passed from the dead S strain, so this dead S strain, to the live R strain, and somehow transformed, somehow changed the R strain. In the 1940s, we had the Hershey-Chase experiments, which concluded DNA is the transforming substance. So they took a virus, they radio-labeled the DNA, they allowed it to infect the bacteria, they agitated to remove the virus, and then they centrifuged and separated, and we can see that now the bacterial sediment is radioactive. So they started with radioactive DNA, and now we see that the bacteria has the radioactive substance inside of it. So somehow the virus had to inject or transform or pass along this material to the bacteria. Just to make sure, they ran the capsid. So they labeled the capsid of the virus. You can see the virus is inserting its DNA, and then they agitated, and then they centrifuged, and we see that the virus particles are still radioactive. So the capsid never entered the bacteria. There was something else, right? And so this is how they showed that DNA is the transforming substance. It wasn't until 1953 that Watson and Crick determined the structure of DNA, and they used other people's research and did not give them credit for it. So if you want to go read about Rosalind Franklin, who was doing some X-ray crystallography studies of DNA, and basically her information was also used to determine the structure of DNA, and she was not given credit for it. So what is DNA? It's a chain of nucleotides. There's always going to be a phosphate, a sugar, and a nitrogenous base. So here we have a phosphate, we have a sugar, and we have a nitrogenous base. So all three of these things together are called a nucleotide. The structure of DNA has four possible bases. There's A, G, T, and C. So there's two purines. Purines have a double ring, that's A and G. And there's two pyrimidines with a single ring, that's T and C. You're going to see that the bases are attached to the sugar and project to the side, and you're going to see how these A and T's and G's and C's actually bind to each other. So here's our complete picture. Here's our phosphate, our sugar, and our nitrogenous base. So you can see here that T only has one ring and A has two rings, right? So two rings are purines and one ring is pyrimidine. And you can see that they match up the same every time. So A bonds to T, and there's two bonds there. G to C, and there's three. And this pattern repeats itself. So I want you to see that the sides of the ladder are always sugars and phosphates, and kind of the rungs of the ladder are always the nitrogenous bases bound together. And these are hydrogen bonds. Remember, hydrogen bonds, lots of them, very strong, but can also be pulled apart and put back together. And you'll see that we need this if we're going to copy DNA. So this base pairing that's always A to T and C to G is called complementary base pairing. Another thing to notice is that one strand is running in the opposite direction, but they're still parallel. So they call this anti-parallel. Here we have an overview of DNA replication. So DNA replication is when DNA is copying itself. So DNA copied to DNA. So you can see you have your double helix strand of DNA. You can see that those hydrogen bonds are broken, and we're going to start the process of copying. So here's our parent DNA, and you can see that those strands split, and then we copy the parent strand, and we end up with these daughter strands. So what's interesting about this, we call this semi-conservative replication. So replication is the process, semi-conservative is the mechanism, the way in which we do DNA replication. What's nice about this is that the parent strand always serves as a template for the newly created strand. So that if any mistakes are made, we have machinery that can come in. So let's say here we put an A instead. When we were creating the strand, we added the wrong nitrogenous base. So we have DNA machinery that can come in and remove this strand. It reads the parent strand and says, oh, it's not supposed to be an A, it's supposed to be a C, let me fix that. So this is a really great process for catching any errors, and this is how we keep the DNA very stable from generation to generation. There are several enzymes responsible for the replication of DNA, or necessary. We have DNA helicase, which is going to unwind and unzip the double strand. Remember, breaking those hydrogen bonds between the nitrogenous base pairs. We also have DNA polymerase. DNA polymerase is going to join new DNA nucleotides. Right? If we're copying, we're building that daughter strand. The DNA polymerase is what joins those new nucleotides to make up that daughter strand. In this image, we can see the DNA helicase. So this is allowing for the unwinding or unzipping of the DNA helix. And then here we have DNA polymerase, which is reading the parent strand and adding the appropriate nucleotide in creating the daughter strand. So of course we have to make this a little bit more difficult because the strands are anti-parallel. So we have the DNA ligase here, and we have our two strands that we've split apart, and we have our DNA pol that's reading. So our DNA polymerase is reading, but as this helicase moves back, the DNA polymerase reads one section. Then the ligase moves back, we've got another section we have to read. So there's always going to be a leading strand and a lagging strand. This is a little difficult because this isn't a moving image. Here the DNA helicase is moving backwards to unwind, right? So it's moving this way and unwinding as it goes. You can see here this DNA polymerase, this is the leading strand because all it has to do is move forward, right? And copy. As the helicase moves backwards, the DNA pol moves forward. But on this strand, to copy this strand, the DNA polymerase is constantly backtracking. So it read this section, then the helicase moved back, now it's reading this section, helicase moved back, it's going to read this section. So you have these disjointed fragments called Okazawa fragments. So this is called the lagging strand. It's not going to link together until the DNA ligase comes in and joins them or ligates them together. So the leading strand, the DNA polymerase is just always moving forward. The lagging strand, DNA pol is binding and then reading, and then it has to bind back here and read, and then it has to bind back here and read. So it's kind of working a little bit harder. Now we move on to gene expression. So when I say gene expression, you should be thinking about DNA being copied into RNA and eventually being translated into a protein. So what we were looking at before, DNA replication, is just if we're copying DNA, like we're doing mitosis or meiosis, and we're copying the DNA before we split the cell. This is the process of how we actually turn DNA messages into proteins. So messenger RNA, transfer RNA, and ribosomal RNA are all important in this gene expression process. So here we have transcription. This takes place in the nucleus. This is where we're taking DNA and we're turning it into RNA, specifically messenger RNA. We're sending a message outside to the cell. So we want to keep the DNA in the nucleus, so transcription happens inside the nucleus. I think back when we talked about cell chapters, I talked about the fact that this DNA is like the reference book you can't take out of the library. You don't want it to leave. You don't want it to go out into the cell to be broken down by digestive enzymes. My new analogy is it's the hard drive at your computer at home. You don't take your hard drive out of your computer and bring it to work and put it into your computer at work, right? You make a copy on a flash drive or email it to yourself and then open it up at work. Translation is where we're going to take that messenger RNA template and we're going to put together a sequence of amino acids and create a polypeptide which will eventually be folded to form a protein. During this translation part, we need transfer RNAs and we also need ribosomal RNA. So you can see the 3 RNAs that are necessary for gene expression. If we have a specific sequence of DNA, we have a gene here. This gene is going to be transcribed into messenger RNA. Messenger RNA is going to be translated into a polypeptide. And what is a polypeptide? I take amino acid 1 and I link it to amino acid 2 and I link it to amino acid 3, and then eventually we're going to fold this polypeptide, and once it's active, it's now called a protein. Since we're taking DNA and transcribing it into RNA, we're going to have RNA polymerase as the enzyme that's going to bind to the section in the DNA and tell it, read here. So a promoter is saying there's a gene right in front of me. You need to read this gene and make the protein product. So you're going to have to open the DNA helix. You're going to have to copy the specific gene, and RNA polymerase is going to join the new RNA nucleotides together. RNA has one base that's different than DNA. So instead of having a T, like DNA, RNA has a U. What's interesting is you automatically know it's RNA. So if I had a DNA sequence that was A, T, G, C, my RNA sequence, it would read the A, and instead of T, it would put a U. T would be A, G would be C, C would be G. So it's complementary base pairing based on the DNA sequence, and instead of T, we have U. Here we have the promoter. The promoter says, just forward of me is a gene. So read here, in front of me is a gene. The RNA polymerase is going to bind, and it's going to start reading the DNA sequence. So if it reads a C on the DNA, the RNA is going to pull a G nucleotide. If it sees G, it's going to have a C. If it sees T, it's going to pull an A. And then the RNA polymerase links all of these nucleotides together. We started with our DNA and a gene, and we've transcribed it into messenger RNA. Before messenger RNA is going to leave the safety of the nucleus, we're going to process the messenger RNA. And this happens in eukaryotes. So we don't have this processing in prokaryotes, right? There is no nucleus. So this is happening in eukaryotes. So we have our messenger RNA, we have our exons, and we have our introns. The introns are going to be removed, and the exon pieces are going to be linked together. So I know it's silly. Introns removed, exons stay. We're also going to put a 5' cap. This helps with ribosome reading. Remember, the ribosomes and the transfer RNA have to turn this messenger RNA into a protein. So that 5' cap helps the machinery read for translation. And then we're going to add a poly A tail, and that's exactly what it sounds like. We link a whole bunch of A's on the end of this messenger RNA, so if some digestive enzyme starts chewing away at the tail, we don't get to the gene coding part, right? And then we're going to see this mature messenger RNA leave through a nuclear pore and enter into the cytoplasm where it can begin translation. Translation is the second step in gene expression, where we're going to create a protein. Remember, we need these 3 items. So here's our mature RNA that just came from the nucleus to the cytoplasm. The RNA, both transfer RNA and ribosomal RNA, these are already in the cytoplasm, and these are created or made from the nucleolus. So you remember, we have the nucleus, and then inside of it we have the nucleolus. The nucleolus is what's going to create the transfer RNA and the ribosomal RNA, and that's all going to be pushed out into the cytoplasm. So you need all 3 of these RNA molecules in the cytoplasm in order for protein synthesis to take place. Here's the big picture, and then we'll kind of go into more detail. I want you to see the messenger RNA is being threaded between the 2 ribosomal RNAs. So we have ribosomal RNA kind of clasping its hands around the messenger RNA, and every 3 letters of the messenger RNA is being read. A transfer RNA comes in, and if there's a match, it drops its amino acid off at the chain. And this polypeptide chain will just keep growing until we get to a stop sequence on the messenger RNA. And then all this machinery will fall off, and hopefully we will have a polypeptide that's good that we can use to fold and create a functioning protein. So the triplet code on the messenger RNA is called a codon. So here's my messenger RNA. Let's say I have A, U, G, C, C. Every 3 letters would be called a codon. There are 64 different messenger RNA codons. There are stop codons. Say, we're done, this is it, we've read the gene, we've made the polypeptide, stop. The genetic code is almost universal in all living organisms. So what we're saying is, if we see messenger RNA AAU, that means the transfer RNA is going to come in, recognize that sequence, and drop off a very specific amino acid. And we see this universally. This is a messenger RNA codon chart. So if the first codon letter is U, The second letter is U, and the third letter is U. Phenylalanine is the amino acid that the transfer RNA is going to drop off. If we have UCC on the messenger RNA, the transfer RNA is going to drop off amino acid serine. And you can see here the stop codons. So there's a chart that you can look all of this up. You don't have to memorize it. There's also a color-coded one in your book. Here we have an image of transfer RNA. So it is RNA, so it's going to be made up of AUs and Gs and Cs, right, nucleotides. You can see they form bonds between nitrogenous bases, and it takes on this kind of shoe structure, some people call it boot structure. And you see the word anticodon. So remember that anticodon is what's going to match the messenger RNA codon. So on the messenger RNA, if you have A, G, C, that's called the codon. On the transfer RNA, you have this sequence, which is the anticodon. The transfer RNA is going to come in and read the codon, and if it's a match, if it's a complementary match, it's going to drop off this amino acid. I do want you to realize that these transfer RNAs are already assembled. So the transfer RNA is created, it goes out, it finds the amino acid that matches its anticodon sequence, and these transfer RNAs are floating around in the cytoplasm ready to be pulled in and drop off their amino acid if the anticodon matches or fits or complementary pairs with the codon on the messenger RNA. So maybe this image makes a little bit more sense now. We have our messenger RNA sequence. Every 3 nucleotides is a codon. We have our transfer RNA, and this is our anticodon, and it's bringing in an amino acid. If the anticodon and the codon match, that amino acid will be dropped off to the growing polypeptide strand. There are 3 steps to translation. We see the first step is initiation. We can see the messenger RNA has bound the small ribosomal subunit. We have our initial start sequence, so AUG is the start sequence on the messenger RNA, and we see that the matching transfer RNA is going to bring methionine amino acid in. We're going to see the large ribosomal subunit attach. We see the complementary base pairing between the transfer RNA and the messenger RNA, and this is happening in the P site. So step 2 is elongation. And remember the previous slide, it said this was E. E stands for exit. This is P, stands for polypeptide, and A stands for attachment. So the new transfer RNA is going to come in. If the anticodon and the codon match, so anticodon on the transfer RNA and the codon on the messenger RNA match, then the polypeptide is going to be transferred to that amino acid and the strand is going to continue to grow. What's going to happen to the P site now? The polypeptide has been transferred, the transfer RNA is in the P site, it's then going to move to the E site or the exit site. So this is kind of like a conveyor belt of things that are happening, right? So you'll usually find the polypeptide in the P site. You'll see that A site is where the transfer RNA comes in. If we have a codon-anticodon match, this polypeptide is going to be transferred to this amino acid, and this transfer RNA is going to go to the exit or ejection site and out. Now these transfer RNAs can go back and pick up another amino acid. So transfer RNAs can be used again and again, but remember the anticodon sequence always has to match the amino acid that's bound. Finally, we're at step 3, which is termination. When we get to the stop codon on the messenger RNA, we're going to have a release factor that's going to bind, and that release factor is going to cause the release of all of these components. And now we have our messenger RNA that's released, we have our polypeptide that's released, we have our ribosomal RNA, and our transfer RNAs. Basically, this process is complete. We read the gene and we made the protein. Here's a nice overview of our gene expression. So we know that the DNA is going to be transcribed into messenger RNA. We know that that messenger RNA has to be processed, right? So we remove the introns. We put together or join the exons. We're going to put a 5' cap and a poly A tail, and we're going to send that messenger RNA out into the cytoplasm of the cell for translation. Then we know that the ribosomal subunits are going to attach. We're going to have transfer RNAs dropping off amino acids to build the polypeptide chain. And eventually, when we get to a stop codon, all of these RNAs are going to disassociate, and we'll have our polypeptide protein.